Hello guys, welcome back to my channel. In this video we are going to talk about cell. The video regarding physiology subjects are available in the physiology playlist. The link is in the description, you can check it out. So let's move on to our topic, studying cell and its structure. So the human body is made up of trillions of cells, and I have done talking about cell in my previous video where I have been explaining the fluids inside the cell ICF and external fluid ECFs. So you can check it out. The link is in the description. Now talking about the body organization, the cell is the smallest part. Group of cells form tissue. Tissue give rise organ. Organ give rise organ system, which makes up the human body later on. So we have been knowing this, we have been recalling this from earlier classes. So most of you might be familiar with this, but if you are not, then there is a simple trick. Like just compare your house with this organization of a human body. Your house is made up of brick that gives rise wall and etc. so on. Okay, so moving on to our topic, that from this discussion we got to know that the cell is the basic unit and is the smallest unit of life of human body. It is the structural as well as the functional unit of the human body. Now we got to know the introduction of cell, but exactly cell karta kya hai? So we will be learning the characteristics of cell. Cell needs nutrition and oxygen for its survival. Second, it produces its own energy. Energy is required for growth as well as repair. Third, it needs waste product like carbon dioxide and other metabolic waste. Fourth, it attacks invader like microorganisms such as bacteria and viruses. Fifth, it maintains medium for its survival. And sixth, it reproduces by division, meiosis, mitosis. But there is exception like neuron cells do not reproduce. Now अब पता चल गया है करता क्या है, अब दिखता क्या है तो we will be studying the structure of cell. So in the diagram above you can see there is a boundary made of brown color, that is the cell membrane. Inside the cell membrane you can see blue dotted fluid, that is the cytoplasm, and somewhere you can see the red dot, that is the nucleus. So in the nucleus and the cytoplasm are called as the cell body. So you can say cell membrane encloses cell body. In the further part of the video would be studying these headlines: cell membrane, cytoplasm and nucleus. Let's start with the first one, that is the cell membrane. I'm denoting it with the capital C and M. So the cell membrane is the protective sheet or the covering of the cell body, you can say like that, and it separates or it creates the boundary between the ECF and ICF. It is also semi-permeable membrane. Only specific substance can move in and out through the cell membrane. The thickness of the cell membrane is around 75 angstrom to 111 angstrom, and it has various names like plasma membrane or plasma lemma. Now, which structure makes the cell membrane? बना किन किन चीज़ों से? तो we will be studying the composition of cell membrane. The first constituent is protein, which makes up 55%. The second is lipid, 40%, and carbohydrates about 5%. So how these three components are arranged to make it look like a cell membrane? So we will be studying the designing and the structure of cell membrane. It's just the introduction part I will be covering here, because to cover the cell model it will make the video huge. So I will covering that part in next video. As soon as I will upload the video, the link will be available in the description. So while studying the cell in the electron microscope, it revealed that it is a unit membrane or three-layered membrane, in which the one is the central layer, that is called as the electron lucent layer, and that appear light in color, while two peripheral layer are called as electron dense layer, and this layer appears dark in the electron micrographs. we see the arrangement of the layer. So you can see the peripheral are denoted in pink, and that are dark color layer and usually made up of proteins, and the central layer is the electron lucent light layer made up of lipid. So what is exactly electron dense or electron lucent? Electron dense, as the name suggests, it will be having the density that prevents electron from penetrating that layer. So as no electron penetrate, it would appear dark in the electron microscope, whereas in electron lucent it will be transparent to the electron, so it will appear light. Basically it is permeable to electron, whereas electron dense layer is not permeable to electron, meaning impermeable. So three models of cell membrane were proposed. The first one was Dennelly-Davidson model. The second was unit membrane model by Robertson, and the third was fluid mosaic model by Nicholson and Singer. And it is the important model and it is most acceptable model till now. So I will cover this in the next video. Link will be made available in the description box. Moving ahead, now let's discuss the composition of cell membrane in detail, starting with the lipid. As you all know, it constitutes 40% of the cell membrane composition. The major lipid found in the cell membrane are phospholipid and cholesterol. The lipid layer is a bilayer structure in the cell membrane. It has fluid type nature, meaning portion of the cell membrane can move along the surface of the cell, as well as the material dissolved in the lipid can move. First, phospholipid. It is the abundant among the lipid. It constituted of two parts, head region and the tail region. The head region is made up of phosphate group, glycerol molecule, and phosphate attached group, or your head group. So basically you can see such diagrams are used to represent phospholipids, like the phosphate group and two fatty acid chain. So in some books you can see a bent fatty acid chain or the kink chain. I will be discussing this whole structure uh, in a while. So the head region here is hydrophilic, meaning water loving, or you can say polar end, because the phosphate is a charged particle, and charged particle can easily dissolve in aqueous solution or water. So it is polar or hydrophilic, because water is not H2, it's breaking ion H plus and OH minus, whereas the tail region is hydrophobic. Phobic meaning water hating or repel the water. So it is also called as the non-polar end. Due to its two properties, polar and non-polar, or hydrophilic and hydrophobic, it is called as amphipathic molecule. Now let's study their arrangement. So you know the cell has water outside it, also the cell has water inside it. So for water outside, the phospholipid will form a monolayer, like its head region is water loving, so it will get attached to the head part, and the tail region is water hating, so it will run away from the water, so it will be on the opposite side. So as you can see in the figure, it will show such monolayer of phospholipid on the ECF side, on the extracellular fluid. Water is also present inside the cell, so for the inside, similar monolayer will be there, just the head will be touching the water side and the tail will be on the opposite end. So combining this both structure, we get the bilayered cell membrane where ECF and ICF are there, and in between we have the bilayer lipid layer where the head is facing outside, meaning the water side, and the tail is inside in between the heads. Now let's study exactly the structure of phospholipid. So in this figure you can see a big purple circle is that is the hydrophilic head, which include the phosphate group and the polar head group attached to the phosphate denoted with R, and there is a glycerol molecule, and the orange part or the orange boxes denotes the saturated fatty acid and unsaturated fatty acid. You can see the unsaturated fatty acid has a bend or kinks. These kinks are due to the double bond. As you can see the double bond there. The common R group can be serine, choline, ethanoamine, and depending on the R group, they are named so. Moving on to our second type of lipid, that is cholesterol. Cholesterol is arranged in between the phospholipid molecules. As you know, phospholipids are soft and oily structure. So these phospholipids arranging with the another phospholipids, these are two oily surfaces, so they cannot be properly packed or properly attached. So to properly pack this phospholipid into a membrane, in between we have cholesterol. So you can say cholesterol is responsible for the structural integrity of the lipid layer. Also, it maintains mechanical stability. I will discuss this part in the factors affecting the fluidity of cell membrane in a while. Cholesterol is also amphipathic in nature, meaning it has both hydrophilic region as well as hydrophobic region. Similarly, it has head, which is made up of hydroxyl group OH, that is polar, or you can say hydrophilic, water loving, so on. Tail region is the carbon ring or the steroid ring, which is non-polar, as well as it has a hydrocarbon chain in the tail region, which is also non-polar, making up the tail region water hating. One more important point regarding cholesterol is that it is absent in plant cell because the cell wall of plant are made up of cellulose. In the next slide, we will study the structure of cholesterol, how it looks. So here you can see the orange part denotes the hydroxyl group, which is the polar region of cholesterol, whereas the long non-polar hydrophobic tail is made up of steroid ring, or you can say carbon ring, along with the hydrocarbon chain. So in the figure, you can see the orange structure. The whole orange structure denotes the cholesterol. So the cholesterol is arranged in between the phospholipid molecule, and it is arranged in the similar manner just like the phospholipid, because the phospholipid and cholesterol have similarities in the structure of polar and non-polar regions. So the polar regions of cholesterol will be attached along the polar region of phospholipid, and the non-polar region of cholesterol will be attached along the non-polar region of the phospholipids. So basically you can say the polar head of the cholesterol appears on the liquid side of the cell, that is outside, and the tail of the cholesterol is inside in between the polar heads. Moving forward, we will be studying the function of lipid layers in the cell membrane. So first, it is semi-permeable membrane, the lipid layer. So only fat-soluble substance can pass through it. Fat-soluble substances like gaseous, oxygen, carbon dioxide, carbon monoxide, ammonium gases, alcohol, fatty acids, cholesterol, different vitamins like A, D, E, K vitamins, hormones like steroid hormones, thyroid hormones can pass through this lipid part of the cell membrane. So the second function is of cholesterol helping peripheral protein by forming high density lipid raft capable of anchoring those peripheral protein. So what is peripheral protein? I will discuss this in detail when we will discuss the proteins of cell membrane. But for now, peripheral proteins are a type of protein present in the cell membrane and that are on the periphery of the cell membrane. So at the end parts of the cell membrane. So it is hard for them to attach because phospholipids are soft and oily. So cholesterol provides them high density anchoring point, high density lipid attaching site, so they can go and attach there. The third point is like phospholipid was fluid in nature, so individual phosphate can move here and there within the membrane, within the bilayer, allowing the membrane to have properties like fluidity and flexibility. This fluidity allows spontaneous breaking and reforming of the membrane, helping in the endocytosis and the exocytosis. Moving on to our next topic, that is factor influencing cell fluidity. The first factor here is environment. When the temperature is too hot, the phospholipid molecules move far away from each other, making the cell membrane too fluid. Whereas when the temperature is cold, the phospholipid molecule come close to each other, making the cell membrane too rigid. You can compare the same things with human. When the temperature is too hot, we prefer staying away from each other so that wind and breeze can pass and we can feel cold. But when the temperature is cold, we prefer staying cozy. Similar, you can compare with the cell membrane also here. But when there is a cholesterol molecule, as you know, cholesterol is in between the phospholipids molecule. So when there is cholesterol, the cell membrane fluidity is ideal because cholesterol helps maintain the mechanical stability and structural integrity of cell membrane as it stabilizes the membrane. Now, second factor that is influence or affected fluidity by the composition of bilayer membrane. So the first is phospholipid structure. So you know phospholipid have head and tail. Tail is made up of the fatty acid. So the fatty acid can be the saturated fatty acid or unsaturated fatty acid. Unsaturated fatty acids make membrane less fluid than that of unsaturated fatty acid. So in the figure you can see the first one is of saturated fatty acid, in which you can see the tail region is neat and uniform. This is because it is the saturated one where there is no double bond or the kings. So it is packed too tightly, making the cell membrane less fluid. Whereas in the second diagram you can see there is unsaturated fatty acid, which is shown by the zigzag line, that implies there are kings or double bond that keeps the phospholipid away from each other and that are not too closely packed, giving rise to their fluidity nature. The second is cholesterol structure. Cholesterol structure acts as a bidirectional regulator of membrane fluidity. At higher temperature, cholesterol stabilizes the membrane and raises the melting point, whereas at the lower temperature, it intercalates between phospholipid and prevent clustering. Hence, I've been saying that it maintains structural integrity and the mechanical stability of the cell membrane. Next topic is regarding protein. Protein makes up the 55% of the cell membrane. Protein are the globular masses floating in the lipid bilayer. Protein molecules are here classified into two categories. The first one is the integral protein and the second is the peripheral protein. Talking about the integral protein, these are the protein that passes through the entire thickness of the cell membrane. As the name suggests, integral, meaning it is integrated with the membrane, or you can say permanently attached or tightly bound with the membrane. also known as intrinsic protein and transmembrane protein, as they span the entire length of the cell membrane. The second is the peripheral protein. As the name suggests, it are present on the periphery of the cell membrane, partially embedded on the outer or the inner surface of the cell membrane. These are temporarily attached by non-covalent interaction, or you can say loosely bounded with the integral protein and the lipid layer of the cell membrane, also known as extrinsic protein. Let's now study the example of integral protein as well as peripheral protein. In integral protein, we have channel protein, hormone receptor, antigen, transporter protein, and some enzyme. Whereas in peripheral protein, we have protein of the cytoskeleton, some carrier or transporter protein, as well as some enzyme. Now let's study the structure of this membrane protein. As you know, in phospholipids, we were dealing with the hydrophilic end and hydrophobic end. Similar things are also here. Here, the amino acids of membrane protein are arranged according to their polarity, meaning non-polar amino acid, that is hydrophobic, water heating, are associated directly with the lipid bilayer, whereas the polar or the hydrophilic amino acids are internally arranged near the aqueous solution. To simply understand this concept, just imagine a hollow cylinder. It will have the internal circular curved structure and the outer circular curved structure. So the outer uh, circular curved structure is non-polar, meaning hydrophobic. So obviously it will attach with the hydrophobic part of the lipid bilayer, whereas the internal part is hydrophilic, meaning water loving, so it will allow water loving things to pass through it, and polar substance can pass through it. Now, how this membrane protein will appear? Firstly, the integral protein. It will appear by three types. The first is alpha helix. Receptor are mostly of this type. The second is helical bundle, like multiple helix are there. Enzyme, receptor, transporter are of helical bundle type. Whereas the third is beta barrel. Channel and transporter appears like this. Now the peripheral protein appearance. They are of basic structures, just like having helix and proteins, carrier, transporter, and some enzyme appears like this. Coming towards the function of protein in the cell membrane, the first basic function of it is like polar substances cannot pass through the lipid membrane, so protein act as a transit and allows these molecules to pass in and out through the cell membrane. So firstly, ions, polar molecule, cannot pass, so uses protein as ion channel, that are tunnel protein, or they uses it as a pump which uses ATP for the exchange. Like consider the example, there is a cell and sodium ion want to go inside, so it will go through the sodium channel because it is the polar substances, and only proteins allow the polar substances to go through it, lipid do not. Second is water, uses aquaporins or water channel. Now you will be saying, like why again channel? Because water is also ions, H plus and OH minus. Third, solutes like sugar, amino acids, uses protein as carrier or transporters. Example, glucose transporter. The fourth function is like water soluble hormone, uses a protein as cell surface receptors. Example is insulin, which is a polypeptide made up of 51 amino acids. Now here you can deal with the two problems. First, it is water soluble, meaning polar substance, and second, it has huge size, 51 amino acids. So insulin will not directly go inside the cell membrane due to its huge size. So there are surface receptor where the insulin gets attached, and this surface receptor acts inside on behalf of the insulin. whereas for non-polar hormones such as thyroid hormone T3, T4, they act directly inside the cell. So the problem with thyroid is there whether the thyroid is present less or more. If it is less, then hypothyroidism, and more, then it is hyperthyroidism. But problem with insulin is insulin not there, type 1 diabetes, receptor of insulin not working properly, type 2 diabetes. Now the last composition of cell membrane, that is the carbohydrates. Carbohydrates make up 5% of the cell membrane. Carbohydrate molecules are present in the form of thin, loose covering over the entire surface of the cell membrane called Glycocalyx. Some carbohydrate molecules are attached to the protein and form glycoprotein or proteinoglycans. Some carbohydrate molecules are attached to the lipid molecule and form glycolipids. The function of carbohydrates in the cell membrane. As you know, carbohydrates are negatively charged, so they do not permit negatively charged substance. Second, glycocalyx helps in the tight fixation of one cell with the another. Third, some carbohydrate molecules function as receptor for some hormones. So the overview of the video, we talked about cell. In that we were discussing the cell membrane and its three components: protein, lipids and carbohydrates. On the screen you can see the fluid mosaic model. Do complete the question and let me know the answer in the comment section. Hit the like button and consider subscribing my channel. Thank you for watching.